हेलो एवरीवन। आज हमारी क्विज है बायोटेक्नोलॉजी एंड इट्स एप्लीकेशन की। स्टार्ट करते हैं हम। चैप्टर खोल लेते हैं अपना। नेट स्लो है। एक तो यार लैपटॉप ऐसा रखने से ना मुझे खुद नहीं दिखता क्वेश्चन। अरे आ गया मैं। आइए आइए क्वेश्चन अटेम्प्ट कीजिए। Question number first is the application of biotechnology include therapeutics, processed food, diagnostic, bioremediation, genetically modified crop, energy production, all of these. All of these is the for first question. Genetically modified crop can be reduced by recombinant DNA technology, somatic hybridization, crossbreeding, micropropagation. Recombinant DNA technology. Second का A is the right answer. Which of the following is not an abiotic stress? Abiotic यानी नॉन लिविंग नहीं है. Pest, cold, drought, heat. Pest. Pest is the right answer. Which of the following cannot be a mean of increasing food production? Cannot be a mean of increasing food production. Agrochemical based agriculture, organic and genetically modified crop based agriculture, inorganic chemical engineering. Inorganic chemical engineering, yes. 4 का 4 है आंसर. Genetic modification cannot cause made crops more tolerant to abiotic stresses, reduce reliance on chemical pesticides, increase efficiency of mineral usage by plant, early exhaustion of fertility of soil. Cannot cause early exhaustion of fertility of soil. Yes, right answer. Farmer of developing world cannot rely completely on agrochemical because they are expensive, don't know how to use, are not trained to use them, of infertile soil. They are expensive. Which of the following is not a critical research area of biotechnology? Providing a best catalyst in the form of improved organism, usually microbes or pure enzyme, creating optimal condition through engineering for a catalyst to act, downstream processing technology to purify the protein or organic compound, upstream processing of all the activated antibodies and plasminogens in the plant body is not a critical research area of biotechnology. Pure enzyme, optimal condition through engineering for a catalyst to act. Last one, I guess. Fourth is the right answer. Which of the following is not an example of biopesticide? BT cotton, BT corn, BT soybean, chili, chili. The Green Revolution increased the food supply to triple fold, double fold, one and a half fold, triple fold. Three critical research areas of biotechnology are providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme, multiple ovulation transfer technology, creating optimal condition through engineering for a catalyst to act, downstream processing technology. Three critical research area of biotechnology are फोर्थ तो है ही सब में हो गया सेकंड भी है third one is creating optimal condition through engineering for a catalyst to act, providing the best catalyst in the form of improved organism using a microbe or pure enzyme. First, second, third, fourth, all. First, third, fourth, second नहीं है. Multiple ovulation transfer technology is not a critical research area. Okay. The increased crop production during Green Revolution in India was mainly due to improved crop variety, agrochemical, better management practices, both B and C. Which of the following is not a reason of less yield by farmers of developing country? Expensive agrochemicals, conventional breeding, existing variety, genetically modified organism. It's not a reason of less yield. Genetically modified organism, yes. The partial reason of increased yield during Green Revolution was use of improved crop variety, use of better management practices, use of agrochemical, use of fertilizers and pesticides, use of agrochemical. Oh, first use of improved crop varieties. The the protein products of the following Bt toxin gene cry1 AC and cry2AB are responsible for bollworm. Hmm. Cultivation of Bt cotton has been much in the news. The prefix Bt means barium nahi, bigger thread nahi, produced by biotechnology using restriction enzyme and ligases carrying an endotoxin gene for from Bacillus thuringiensis. Bt toxin does not harm tobacco bedworm, armyworm, beetles, bacillus. Bt toxin is harmful to insects like Lepidopteran, tobacco budworm, army worms, coleopterans, beetle, dipterans, flies, and mosquito. All are correct. In Bt cotton, the Bt toxin present in plant tissue as protoxin is converted into active toxin due to alkaline pH of insect gut. What is true about Bt toxin? The inactive protoxin get converted into active form in the insect gut. Bt toxin exists as active toxin in the bacillus. The activated toxin enters the ovary of the pest to sterilize it and thus prevent its multiplication. The concerned bacillus has antitoxin. Is the right answer. Bacillus thuringiensis strains have been used for designing novel bioinsecticidal plants, biomineralization processes, biofertilizer, biometallurgical techniques. First is the right answer for this. The activated BD toxin binds to surface of midgut or epithelial cell, surface of hindgut, foregut, gut endothelial cell. Midgut. Yeah, hindgut. Midgut. Midgut. The bacterium Bacillus thuringiensis is widely used in contemporary biology as an indicator of water pollution, insecticide, agent for production of dairy products, source of industrial enzyme. Is widely used in contemporary biology as an indicator of water pollution, insecticide I guess is the right answer. 22 का 2 is the right answer. Perithoragenesis produce protein dash during dash their life cycle. Crystal during specific phase of life cycle. Yes, सही है. Which of the following lepidopteran is killed by protein produced by a particular strain of Bacillus thuringiensis? Mosquito, flies, beetle, tobacco, budworm. Fourth is the right answer. Most of the Bt toxin genes are insect group specific, pest specific, weed specific, bacteria specific, insect group specific. Cryotoxin acts by the lysis of intestine cell binding to midgut epithelial cell swelling. More than one correct. First and second. More than one correct. Cry-1 endotoxin obtained from Bacillus thuringiensis are effective against nematodes, bollworms, flies, bollworms. The choice of Bt toxin gene depend on crop targeted pest, both A and B, only on protein of Bt toxin gene. Choice of Bt toxin gene depend on crop and targeted pest. Yes. Corn borer are characterized by Cry 1 AC, Cry 1 B AB, Cry 2 AB, both B and C. Cry 1 AB. 29 का फर्स्ट इज द राइट आंसर. पुष्पेंद्र जी, चिराग जी एंड कीप गोइंग तो खुद ही नाम ही कीप गोइंग है. कीप गोइंग सब लोग. BD toxin gene is expressed in plant, fungi, algae, bacteria. BD toxin gene is expressed in plant. Which of the following Coleoptera is killed by protein produced by a particular strain of Bacillus thuringiensis? Mosquito, flies, beetle, tobacco, budworm. Oh, beetles. Bacillus thuringiensis for beetles. Which of the following event is second in the action of Bt toxin? Binding to epithelial cell, creating pores, swelling, lysis. ओके, दूसरे पहला तो एक्टिवेशन हो गया, दूसरा बाइंडिंग. देखते हैं. नहीं, दूसरा इज क्रिएटिंग पोर. Some of the characteristics of Bt cotton are long fiber and resistant to aphids, medium yield, long fiber and resistant to beetle pest, high yield and production of toxic protein crystal which kill dipteran pest, high yield and resistant to bollworms. High yield and resistant to bollworms is the right answer. What is an antisense technology? When a piece of RNA that is complementary in sequence is used to stop expression of a specific gene. Third, RNA interference inhibit DNA replication, neither transcription nor translation of specific gene, transcription of gene translation of specific mRNAs. Which part of tobacco plant is infected by maladjustinine incognitia root? PYQ have already done. Which of the following is commonly used in transfer of foreign DNA into crop plants? Agrobacterium tumefaciens PYQ again. RNA is a mechanism to silence gene with the help of double-stranded RNA. 38 ka 2. The process of RNA interference has been used in the development of plant resistance to fungi, virus, insect, nematodes. Select silencing of specific mRNA due to contemporary double-stranded RNA is called mRNA silencing. Here, the source of double-stranded RNA is RNA virus, mobile genetic element with RNA intermediate, transposons having RNA intermediate during process of replications. All of these. All of these? Yes, all of these. Agrobacterium vector carried nematode and nematode specific gene which is introduced into the host plant and such that it produces sense and antisense protein. No. Sense and antisense RNA. Oh, yeah. My bad. RNA interference is essential for the cell proliferation, cell defense, cell differentiation, micropropagation, cell defense. From RNA interference technique, toxins are made to kill soil parasite, made crop more tolerant to abiotic stress. Plants are made resistant to soil parasite. Plants are developed to get the enhanced nutritional value of food. Toxins are made to kill soil parasite, made crop more benefit to abiotic, made resistant to soil parasites. Yes, 43 ka 3. Select the correct statement. RNA interference is a post-transcriptional gene silencing method. Transgenic tomato have been developed to reduce pre-harvest losses by decreasing their shelf life. In GMO, early exhaustion of fertility soil is promoted. Gene Cryvan AB control infection, maladjustinine incognitia in tobacco plants. By decreasing their shelf life. First, RNA interference is a post-transcriptional gene silencing method. When DNA is transcribed into mRNA, usually the mRNA remain single-stranded, but in some cases, RNA can be made that is complementary to the mRNA. This is called antisense RNA, and its main function is to block gene expression, amplify mRNA, enhance first. To protect tobacco plant against maladjustinine incognitia using RNA interference, nematode-specific gene were introduced into the plant using Agrobacterium tumefaciens. RNA interference is a method of gene silencing which, when done in tobacco plant to prevent infestation by maladjustinine incognitia, may involve all expect bacteria. Agrobacterium as vector introduction of single-stranded RNA into host cell Transposons that replicate via RNA interference silencing of mRNA to stop translation. Yes, 47 का 2 is the right answer. Consider the following statement: RNA interference take place in all eukaryotic organisms as a method of cellular defense. As a method of cellular defense, it involves the silencing of specific mRNA due due to complementary double-stranded DNA molecule that prevent translation of mRNA. Fire and Mello got the Nobel Prize for the discovery of RNA interference. Second is wrong. First and third. Fire and Mello got the Nobel Prize for the discovery of RNA interference. This is a new statement for me. Which of the following genes do not occur naturally in living organisms? Bt genes, RNA interference genes, cry genes not occur naturally. Endogenous cytoplasmic defense genes. Last one. Consumption of which one of the following food can prevent the kind of blindness associated with vitamin A deficiency? Gordon rice. 50 का 3 is the right answer. 50 क्वेश्चन हमारे हो गए हैं कंप्लीट. ये PYQ है इसलिए ऑलरेडी सिलेक्टेड है. और आज चैप्टर भी इजी था प्लस थोड़ा प्रेजेंस ऑफ माइंड भी था तो कम ही गलत हुए. आप लोगों का क्या रिस्पॉन्स रहा मुझे बताइएगा. बाय बाय. लाइक शेयर सब्सक्राइब माय चैनल एंड थैंक यू फॉर वॉचिंग.